The second method is chemotherapy or drug therapy. It is a method where use of certain of drugs to treat cancer is seen. Drugs are generally injected using the circulatory system. Sometimes two or more drugs are combined to have better effect in the chemotherapy. For example, antimetabolites such as mercaptopurines, antibiotics such as actinomycins, alkylating agents such as carboxyamides, mitotic inhibitors such as vincristines, hormones such as estrogen and cortisones are widely used in chemotherapy or drug therapy. Chemotherapy is given at regular intervals, which we call it as cycles. A cycle includes dose of drugs for a period of time. Between the cycles, there is a period of rest which is used to observe the side effects of chemo drugs. There are cancer vaccines that are used to boost the immunity against the infectious retroviruses. This method is also called as biological therapy or immunotherapy. Coming on to the advantages, chemotherapy is very effective and kills the cancerous cells. It causes shrinkage of tumors. Disadvantages is it includes many side effects such as it also kills normal and healthy cells of the body. There will be loss of hair due to destruction in the hair follicles. There will be diarrhea caused due to the destruction of cells of intestinal lining, and mostly since it is administered through the blood system, it causes damage of blood cells. The third method we have is gene therapy or gene augmentation therapy, GAT. As cancer is due to mutation in gene, introduction of extra copies of normal genes into the body of a cancer patient helps in the production of a normal gene product. So, the cell which is cancerous will behave as a normal cell. There is a popular cancer gene therapy method called target killing specific cells. In this method, genes are directed to the target cells and then expressed so as to kill themselves. So self-killing or suicidal process takes place in this method. It is achieved by the introduction of a lethal toxin production or producing gene. This method is generally called as target killing specific cells. There is another method called target inhibition gene expression method. In this gene therapy method, expression of cancer causing genes is blocked at different levels of their function, so that gene products are stopped from its production and they fail to act upon the cells. Gene therapy has two ways or two methods of approach: ex vivo gene therapy, in vivo gene therapy. In ex vivo gene therapy, transfer of gene takes place. Initially, transfer of cloned genes are grown outside the body of the patient in the laboratories by using cell culture. Successful genic cells are then selected and introduced into the patient and subjected for the process of engrafting. So, in ex vivo method, target production of gene is seen in the cells which are cultured outside the body of the patient. The second method is in vivo method. In this gene transfer method, the cloned genes are transferred directly into the tissues of the patient. It is by using either liposomes or sometimes even viral vectors. it is used widely to treat inner organs and vital organs such as liver cancer and brain cancer, etc. Coming on to the advantages, it is a positive medical outcome of killing cancerous cells is achieved by gene therapy process. Gene therapy process has less or no side effects. It has less or no pain in the treatment process. And once if it gives a positive result, the permanent cure of cancer could be seen. Last point of disadvantages is it affects the immune system. It sometimes targets the wrong cells and not healthy cells. Viral vectors which are used may trigger certain of diseases in the individuals, may even develop the resistance for the gene therapy, and it is highly expensive method, and there is a clearance needed from the ethical committee. So this is about the gene therapy. So radiation therapy, chemotherapy, gene therapy are the three major methods which are used to prevent and regulate the cancer in cancer patients.